This Week in Virology, the podcast about viruses, the kind that make you sick. From MicrobeTV, this is TWiV. This Week in Virology, episode 1307, recorded on March 20, 2026. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It's not a bad day in Madison. It's 63 Fahrenheit, which is pretty nice, although it is a bit overcast. But I'm happy to see a little bit of warmth.
It's 15 C here. That's like tropical compared to what it's been.
Exactly.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. If it's 16 there, that means it's 61 Fahrenheit.
There you go.
Yeah. So here it's 68 Fahrenheit and it's 20 degrees Celsius and blue sky and not any clouds. It's just really nice. 68 degrees. Wow.
What just happened in Michigan of note? Something happened. Like some weather-related thing? Or
Oh, no, Cleveland. That's my pick.
Okay.
But
I didn't see that.
Yeah.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 88 degrees and sunny. What can I tell you?
It's
Austin, beautiful.
Texas. Oh, if you enjoy these programs, we would love your support. Go to microbe.tv/contribute. We were just complaining before we started. And, you know, when it has to do with viruses, we do it on the program to each other, right? But when it's not viruses, we don't want to record it and you don't have to hear it. But it's the same idea, right? I had something on my mind I wanted to get off, so I was complaining to everyone. And then after they got quiet, I knew that was enough. It's time to start talking about viruses. But if it's a virus thing, I rant here because you have to know about it. The Rosenfeld Lab is looking for a postdoc and a technician. They have moved from the FDA to Philadelphia. They work on antiviruses. They're interested in questions about pathogenesis. They're making animal models. They want to know why they induce, or what's the function of cross-reactive antibodies that are induced, or what's the effect on pathogenesis. And they're working on new vaccine platforms. So if you are interested, email is rosenfelda@mlhs.org. So Kathy, we haven't talked about ASV in a long time. Do you want to say something?
Sure. ASV 2026 will be July 27th to 30th in Minneapolis at the University of Minnesota. And the full program is now available in a drop-down tab, ASV 2026, if you go to the website asv.org. So in that tab, then you can see things about the program, registration, and so forth. Scientific highlights include the fact that Steve Goff, Columbia University, is going to be the keynote speaker. There will be talks from the Ann Palmenberg Junior Investigator Awards, and there will be 5 satellites in the areas of animal virology, evolution and ecology, research experiences in industry, prokaryotic virology, and from the global scholars. This one will be entitled Virology in One Voice. And once again, childcare will be available on site to ASV members through Kitty Corp. And so there's information about that in the drop down. Also, there's other support options from ASV for child and dependent care through grants from ASV called ASV Cares. And those deadlines and information about that applying for ASV Cares will go out with the abstract information around the end of March. So that information has taken a while to get propagated in the new website. And because the meeting is pretty late in July, you have a while before early bird registration happens, and that won't be until June 1st. And there will also be digital access available for the meeting, and details are in the registration tab. So a few more updates for you to think about as you plan your summer attendance at meetings, and the ASV meeting is going to be a really good one.
So digital means you can do remote?
Digital means that you can do live stream and later recordings of
cool.
some of the things. Of course, not the individual talks, the 15-minute
Yeah.
talks and the special workshops and things like that.
But I guess keynote and plenaries, right?
Correct. And I
So-
believe the satellites. It's all spelled out on that website.
It's
So
been
is,
that's what it's been in the past,
yeah.
It has?
Yeah.
Okay.
and also
That's
the
good.
state of the arts.
All right. We have a little news for you. Someone put the links in. Who would like to talk about these?
I put the link in for the obituary for Robin Weiss.
Did you know him?
I did know him.
Oh, tell us then.
Actually, I didn't know him well. We overlapped at the Imperial Cancer Research Fund. He was there, I suppose, in a professorial capacity while I was there as a postdoc. So I knew who he was. I heard him give a couple of talks, etc. I doubt if he knew who I was, a lowly postdoc. But he was a retrovirologist to start with, who evolved over time through the ICRF, Imperial Cancer Research Fund, became the Institute for Cancer Research in England, and I think he even directed that for a while. All the details are in this Institute for Cancer Research obituary for him. But he made some seminal observations in HIV biology, including, I think he identified, according to this, the CD4 as a receptor for HIV. So he was quite a respected researcher, and we're sorry to see him go. He was 86 years old. The other little clip I put in here was actually somebody put in the NPR link. I've got a link to a New York Times article that many people are probably familiar with a federal judge responding to lawsuits issued by a number of different medical organizations challenging RFK's vaccine policies, and the judge ruled that they are basically unscientific and cannot be enforced. Okay, so he I don't know legally how you do this, but the title is that they are stricken down. Severe blah blah blah. Federal judge in Massachusetts on Monday blocked the government from implementing a series of decisions on vaccines made over the last year by Health Secretary RFK Jr. So the ruling reversed, at least for the time being, all decisions made by the panelists that Mr. Kennedy appointed to the Advisory Committee for Immunization Practices, which makes recommendations on vaccines. The court decision will prevent the committee from meeting later this week, as it was scheduled to do. You can imagine that this will be challenged, and this will go on forever. However, it is from my perspective at least, a welcome legal challenge to RFK's vaccine policies.
It's great. It's great. He just is a cowboy. He thinks he can do everything on his own. But these committees are supposed to be picked in a certain way. There are federal guidelines, and they just didn't abide by anyone, So they just keep losing in court because they're doing things that are wrong. So I read this morning that HHS is considering whether to appeal or whether to just reconstitute the committee. Can you imagine? I mean, I can't believe that they will have bona fide experts on that committee.
By the way, I don't think I picked this, but I'm for a separate reason. I'm putting together a talk on vaccines, and I would advise people that the leaders at this point in maintaining a rational vaccine policy is the American Academy of Pediatrics. So if you go to the American Academy of Pediatrics website, you can find the old immunization, the past immunization schedule, and a lot of information about it and the changes that have taken place. And I should point out that the American Academy of Pediatrics is supported by at least a dozen different reputable medical organizations in maintaining the old guidelines. So there are resources out there. I would also point out that all of this stuff, CDC or AAP, whatever, these are recommendations. They're guidelines. They're not rules. If there are any rules imposed, it's done at a statewide level. However, if there's, the recommendations cast a huge influence on what the states adopt and, what people perceive they might or should do. So they're important. And if it were me, I would go to the American Academy of Pediatrics.
I just hope that people do that because it's confusing to people, right? To have it change and then not change.
Mm-hmm.
But and they're fighting it, right? Which is not right. That the judge did the right thing. You need to have experts on these committees. And RFK Jr. has said we don't need any experts. We can just go by our feelings. Gosh, what kind of an era are we in? Okay.
The thing that drives me crazy is that, and this seems to be the pattern in this is sort of a paradigm in this administration, whether it be healthcare or whatever, is that you simply make these promulgations, executive order or whatever, okay? And wait for it to be challenged. And the problem is that the legal challenges take forever, in particular when there's appeals and et cetera. And in the meantime, you do whatever you can to implement your whatever policy you want. In the long run, if the legal challenges prevail, now you have to undo all of the stuff that you did, and it just creates a chaotic mess. It's just crazy. I mean, there's a reason. Government is slow, sure, but there's a reason for this. Okay,
right.
It needs to be a deliberative process.
Well, and all of that chaos that Rich mentions just sows distrust in the whole thing and makes people kind of want to throw up their hands about the whole business.
Yeah.
The part of the thing that's being implemented in the new vaccine policies is this concept of shared clinical decision making. SCDM. When I saw that, I thought, oh, okay, so is this some new thing, all right, that we're using to sort of help people feel empowered and feel like they're not being over-mandated? Turns out, no. Shared clinical decision-making is a concept that's been around for a while. It makes perfect sense if you're doing something like trying to figure out how you're going to treat your cancer, right? But for some other things, like shared clinical decision-making on whether or not you should give your child a birth dose of the hepatitis B vaccine, in fact, doesn't make any sense at all, okay? Because we've essentially tried that already, and it doesn't work because people slip through the cracks.
Yep. Medical, what is it called? Medical freedom.
Yeah.
Oh, well, we could go on and on, but let's do some papers.
Yay, data!
We have two very interesting papers today. The first one is a Cell Report paper called Structural Basis for Rep-mediated adeno-associated virus packaging. And the authors are Victoria Xu is the first. Then we have McInnes, Wake, Acebron, Garcia de Ulate, Barrett, Bubeck, and the last corresponding author, actually there are two, Rouse and Barrett and Bubeck are three corresponding authors. This comes from Imperial College London and AstraZeneca in the UK. And Kathy, you want to give us a summary?
Sure. So adeno-associated virus packaging is the topic. AAV is what we call adeno-associated virus for short. And AAV is used in gene therapy. And how its single-strand DNA gets packaged is the topic of this paper. It's significant because it's both important for fundamental virology and also because it can improve the efficacy of AAV gene therapy vectors. And so I'll try to be brief. I think others will want to go into more detail, but AAV has a very small genome, and so the virus depends on the host cell and products of a co-infection by another virus to complete its life cycle. And oftentimes that other virus is adenovirus, hence its name, adeno-associated virus. For example, it uses the host DNA polymerase for viral DNA replication. But the AAV virus encodes a protein that's really crucial for several steps of DNA metabolism, and it's called Rep, and it's in the title. There are 4 isoforms, Rep 78, 68, 52, and 40, and I think those have to do with molecular weights.
Yeah.
Okay. And they all have helicase activity. So what's a helicase? Well, it's a motor protein that separates double-stranded nucleic acids into single strands and using energy from ATP hydrolysis. So the first 2 isoforms of Rep nick and unwind the DNA during replication, and then the smaller reps, 52 and 40, package DNA into empty capsids. And this latter step is what's investigated in detail here. So they use cryo-EM, cryo-electron microscopy, which I'm not sure we've talked about a whole lot recently in detail, but basically samples are flash frozen, ultra rapidly cooled, and that minimizes damage and enables high resolution 3D imaging. And they find what to me is the most remarkable thing. I saw it right away in the graphical abstract, and then I went through and found another figure in the paper, and it's open access, so you can look at these. And so first I have to give you a little more background. There's the empty capsid is where the DNA is going to get packaged into, and it's icosahedral, and so it has 12 vertices that are pentameric, so they're at the 5-fold axis of symmetry. And so there's a complex of 5 Rep-40 molecules that is a pentamer, in other words, 5 subunits, and it binds to one of these pentameric vertices of the empty capsid. Actually, it can bind to all of them, but the authors surmise that the first one that it gets bound to is the one that it just proceeds with, takes priority. And then there's another complex of 7 Rep-40s, and that is, it's a heptamer, hepta meaning 7, and it associates with the viral DNA. And as we'll get into, the viral DNA has a little bit of double-strandedness called the inverted terminal repeat, ITR, and that serves as the packaging signal. Anyway, so this complex of 7 Rep-40s docks, well, first of all, so the pentamer serves as a docking platform, and this pentamer of the reps bound to the empty capsid serves as a docking program for this 7-mer complex that also has the DNA. And so here's the really cool thing. Once this 7-mer docks onto the 5-mer, one of the subunits shuffles to the pentamer from the 7-mer. So now there's 2 hexamers, 6-mers. And simultaneously, this subunit loss from the 7-mer triggers melting of that little bit of double-stranded DNA, unwinding of it, so that it now allows the single-stranded DNA to get inserted into the empty capsid. So they show all this, this is their model, and they show all kinds of figures that support this model. They also identify a cryptic ATP binding site on the Rep-40 that's bound to the AAV8 capsid. So sometimes they use AAV2 and sometimes they use AAV8. So as I said, the paper has a really nice model in the graphical abstract and also in the final figure. There were a couple of terms that I wish they could have introduced a little better, and maybe my colleagues will help us with that. Passive strand versus tracking stand, the AAA plus domain, the Rep protein's ATPase cycle. Most of the I could discern from the context. And they also talked briefly about a latch linker. But then they talked about one concept, which was really easy to get, but which I have never seen before in a scientific paper. Dimers switch sides like a paper fortune teller. So those are those things that go like that. Cool. Anyway, so I thought that was cool. Pretty cool. Let's see. And they have a really nice video that kind of shows that paper fortune teller-like motion. And they show a movie that shows this overall model of the 7-mer binding to the 5-mer and then the subunit transferring and the DNA getting packaged. So I had to watch those supplemental videos on the journal website itself within the paper's text. For both papers, I found finding the supplemental videos and figures a little bit Byzantine, but okay. So that's all I have about background.
The video is just awesome and really, it really wraps the whole thing together. I would have one, I don't know if it's a clarification or my own personalization of, because I have some experience with helicases, having worked on a protein with a helicase domain for a long time. And these proteins were discovered as, or things with helicase, what we call helicase domains now, were discovered as proteins that unwind double-stranded DNA. But the activity is more general than that. Okay, these are generally things that bind to nucleic acids, some single-stranded, some double-stranded, and move. Okay, so it's more appropriately in a way called a motor protein, even though the domains are called helicase domains. And just as an example, the lambda rho termination factor. Okay, it binds to RNA that's, it's a transcription termination factor. It binds to RNA that's coming out of the RNA polymerase and basically motors along the RNA towards the polymerase and rips the RNA out of the polymerase. And that's how the termination happens. So in this case, the helicase is stuffing single-stranded DNA into the capsid. I was not aware of this, but if you look at the literature, this notion that, I was not aware that Rep had to do with DNA packaging. That's just because I'm uninformed. That goes back 25 years.
Mm-hmm. Yeah. Yeah, so this is a motor. They say AAVs use Rep motors to package DNA. And I did not know that either. And I teach AAVs, so I was uninformed also. So these are vectors. These are great vectors for gene therapy. There are 7 FDA EMA-approved AAV-based gene therapy products already. And one of them is for blindness. Lux, what was the name of the drug? Lux something. You inject it into the retina and it resolves some kinds of blindness. And then there are 230 clinical trials worldwide with these viruses. They're nice because they're easy to manipulate and you can get long-term gene expression. They don't integrate, but they remain episomal for a long period of time, which was unexpected. And so they're very useful. But the reason they're doing this work purportedly, I think it's just really interesting, but the reason is that packaging is very inefficient. And so maybe if you could make it better, that would be good to maybe make these motors more efficient. And so we don't really understand how that works. So that's what they're doing in this paper. So they insert the single-stranded DNA into empty capsids. So the empty capsids get assembled first, and then the DNA is inserted by these motors. Yeah, there you go. There's the icosahedral capsid.
My icosahedron earlier was squished, so I had to fix it.
The DNA is single-stranded, but the ends have inverted terminal repeats, which form these double-stranded T structures, which are primers for DNA replication. So the primer for DNA synthesis is built into the genome. It's very clever. It avoids the end problem. And then the Rep proteins, the 4 of them are really important. They are important for nicking the initial double-stranded product. So you get a 3' hydroxyl for priming. They recruit the cellular DNA synthesis machinery to the viral DNA, because otherwise, you have this viral DNA floating around in the nucleus. Nobody's going to pay attention to it unless it's got this Rep protein that's pulling the DNA machinery. And then the Rep proteins are helicases with ATPase activity that pump the DNA in. And so they want to do the structure of this. How does it work? So they use cryo-electron microscopy to visualize the structure of Rep40 in its different forms with and without the capsid. And so the initial analysis, the cryo-EM of Rep40 together with the ITR shows that it forms both heptamers and hexamers, as Kathy said, when it's bound to the DNA. and the structures show how the ATP cycle allosterically affects gripping of the DNA, right? So the DNA is pumped in by this motor. And so ATP binding triggers the compaction of this AAA plus domain, which is, as Kathy said, they don't really talk about it. But mice, I think it's part of the ATPase domain, right?
I think
That's
so
what
too.
that's what I read into it. It causes a compaction, which positions some loops of the protein for DNA binding. And then in the heptamer, double-stranded DNA complex, the tracking strand, I don't know what that is either, is engaged by specific loops. And then an ATPase cycle starts where there are lots of movements in the protein, and that leads to hydrolysis of ATP. In the hexamer, the single-stranded DNA is engaged by they call these pore loops in the protein of all the 6 subunits, and then the ATP is held for 4 steps before being hydrolyzed at the 5th step. And so these show how Rep-40 basically tunes its oligomeric state for either a double-stranded or a single-stranded region for melting and translocation. It's very cool. It's beautiful, really. Then they want to understand how ATPase impacts binding. There are a lot of details that I'm not going to go into, but basically they show that the heptameric Rep-40 binds double-stranded DNA, and the parts of the protein, which are called pore loop wedges, deform the minor groove of the DNA, and that's going to eventually denature it. It's very cool. Now, the hexameric Rep-40 has a narrow lumen and uses all 6 sets of each subunit has pore loops. So this uses all 6 sets of pore loops for single-stranded DNA binding. And they make a helical arrangement around the tightly wound single-stranded backbone. Now, these helicases are dynamic, right? They're not static. And the cryo-EM are just static images. So they want to know what's going on. So they made structures. They classified the images that they got into different states because they're all different in there. And they can see motions going on. So they can see this AAA plus lobe rotating upon release of ADP. So apparently on ATP hydrolysis, it triggers dissociation of DNA, and then ATP rebinding allows DNA to bind at the next base pair. So it's basically one base pair at a time per ATP hydrolyzed. It's pushing it into the capsid, right? And one ATP hydrolysis pushes one base further towards the capsid, and they can see those states in their structures. It's just great. I really like it. They also look at melting, how the protein melts the DNA, and basically it's a multi-step melting mechanism. Step 1, the heptamer docks onto the double-stranded DNA and slightly distorts the duplex. Then it loses one subunit, as Kathy said. That constricts the pore. 3, then you have ATP-driven compaction of the nucleotide-bound subunit, which separates the strands. And then 4, the hexamer now drives migration of the protein through the passive strand. And so basically that's how the DNA is denatured, and because it has to be single-stranded to get into the capsid. So how does it actually get it in? So Rep-40, they solved the structure of Rep-40 together with the capsid now, because so far we've got it Rep-40 with DNA. And they get a 1.9 angstrom resolution. And what's cool is that 94% of the 5-fold axes are occupied by Rep-40, right? So as Kathy said, Rep-40 can bind every 5-fold axis. So they're indistinguishable initially, right? And what's also cool is that it preferentially binds empty capsids, which makes sense, right? Because you don't need to do this with a full capsid because there's already DNA in there. So that's great that it does that. I don't know how it does that. I don't think they know how it does that, but that's
well. They speculate. You're probably getting to this, but they speculate ultimately that in order to, you really only need one of these machines per capsid to work, that when one binds, there's conformational changes in the capsid that exclude the binding of the others. And I would assume that a full capsid has subtle conformational differences from an empty capsid that this protein can distinguish.
Yeah, I was sort of assuming they're probably, because I was imagining, well, what if there are these complexes on multiple 5-fold axes? How do you make sure that you don't, load at 2 of them at the same time, package at 2 of them at the same time? And so my assumption was that when one started to actually have a pentamer and heptamer bound, there was some sort of subtle conformational change that removed the others.
That's reminds me that the typical sort of out, the hand waving out in almost anything is, oh, there's a conformational change.
Yeah.
So what do they find? They have the capsid bound to Rep-40. So Rep-40 binds via this AAA plus domain. And that seems to drive pentamerization, right? So 5, they're beta hairpins in Rep, and 5 of these insert between the capsid protein of the virus that forms the 5-fold pore, right? So they're sticking into the capsid, and it's stabilized. They can see exactly how this interaction is stabilized. This also twists the loops of the capsid, and so there's a pore at the 5-fold axis where this is happening, and the twisting constricts the pore and widens the base of on the capsid, right? At the same time, they say the interior electrostatics are remodeled. You get a new basic patch at the rim, and they think that has to do with DNA funneling, right? Because the DNA has a different charge. So basically, the insertion of this Rep-40 beta strand into the 5-fold pore, it primes it for packaging by widening the base of the pore and changing the interior electrostatics, and it constricts the pore tips. And they think that deters non-specific DNA threading. Other DNAs can't get in because of that. It's just amazing, right? The binding of the protein to the capsid does all of this thing. Okay, so then they compare the structures of AAV8 alone with Rep-40 bound AAV, and they can see differences, right? As Rich just said, and there are loops on the empty capsid that are unbuckled. When Rep-40 binds, these loops unbuckle. They flip away from the capsid, and they weaken it in a certain way, and they can see again exactly the interactions that are going here. There's also a cryptic binding site on the 2-fold plateau of the particle that's bound to Rep-40. And so they're wondering if ATP is an allosteric modulator of the capsid. So the ATP binding site is on the capsid, and when it's available when Rep-40 binds to it. So Rep-40 and this ATP-ADP triggers additional rearrangements on the interior. So basically, to summarize it, Rep-40 alters the capsid pore size, seals the entrance so foreign DNA can't get in. Because remember, now we have this Rep protein bound, and the DNA is threading through it, so the pore doesn't need to be big anymore. It constricts just to accommodate that DNA. And then there are the nucleotide binding triggers structural changes that prime the capsid for the packaging reaction. I mean, they don't go beyond that, right? They don't watch it go in, but presumably you could do that as well. So the cool thing here is that the Rep-40 has different oligomeric states, one for the helicase and one for the packaging, right? So here's the model. This is just amazing. So the pentamer of Rep-40 binds the capsid. This is now the docking platform for the heptamer that's bound to the double-stranded part of the viral DNA. When these associated, as Kathy said, the heptamer puts a subunit into the pentamer. So now we have 2 hexameric capsid-anchored motors, and a subunit loss in the loss in the heptamer. It triggers melting of the DNA, and that initiates the insertion of the single-stranded DNA into the capsids. And they say, you know, all these. So AAV doesn't have a portal protein. Herpes viruses, on the other hand, herpes simplex, has a portal at one 5-fold axis that serves for DNA entry and exit. But most viruses do not have portals. And so they say for all these icosahedral viruses where there are 5-fold axes of symmetry, every site is a potential packaging site. But what they say and show here is that once the first active motor forms, and it could form anywhere, it inactivates the remaining pores, and that's what they get hints of from their structures.
It's basically assembling a portal on one of the
Yeah.
vertices.
Exactly right. Exactly right. So they say our discovery of a cryptic ATP binding site on the Rep-bound empty capsid illustrates the genetic economy of this minimalistic virus. It really is. It's a small genome. It's a couple of proteins. Rep and cap. That's it. It's really cool. So now they think they can engineer these things to be better. Now, this got me thinking about other viruses. So for poliovirus, when it binds its receptor, a portal forms, or it's called an umbilicus, that goes between the virus and the membrane, and the RNA goes through it. It's not at the 5-fold axis. It's at the 5-fold axis. The initial binding is at the 5-fold axis, but the portal forms in a different place. And so you could imagine the same thing happening. Maybe every 5-fold axis is amenable to binding, but once one forms, maybe the others are deformed. And that could be looked at now that you have these clues here. The difference is, for poliovirus, it's been shown, for picornas, it's been shown that the 3' end of the RNA goes out first. This is on exit, not packaging, because we don't even know how packaging works. So how does it know? Or is it just random, right? You got 12 chances to hit the 3' end, or maybe more than 12. I don't know. Maybe you get one, it works, or it doesn't work. If it doesn't work, it's a non-infectious particle, right? But
I'm gonna guess it's not random. That's one of the things that impresses me about this paper, is that it's a very deliberate, if I can use that word, a very deliberate process. It just boggles the mind.
Yeah, I like in the discussion how they talk about docking a heptamer to an active hexamer transition has been reported for several other helicases. So bacteriophage T7 and a mitochondrial helicase that's called Twinkle. But then they talk about how SV40 T antigen and papillomavirus, but T antigen, the one I'm most familiar with, uses double hexamer for origin melting. So, so the AAV is getting to that double hexamer morphology by this transition, and then functioning as a helicase. I don't know. They just did a nice job of comparison to other viruses and what's known about helicases.
Yeah, for some reason, I was struck, and this is very strange, by the ATP and the idea of the one ATP to move one base pair into the capsid, and sort of doing out the back of the envelope math of how much ATP is used just for packaging AAV in one infected cell, and getting huge numbers like 5 million ATPs or something like that. And sort of, thinking like, wow, when I talk about ATP production in other aspects of metabolism, those 32 ATPs are nothing.
Oh, yeah. Niles used to tell me this all the time. Don't worry, there's plenty of ATP. Not a problem.
It takes 2 ATP to make a peptide bond.
So I have a couple of thoughts. First of all, embedded in here somewhere, and it's not even embedded, it's right there, but I didn't have the patience to pursue it through. I would need somebody to guide me through this, is actually in detail mechanically how the ATP hydrolysis makes all this happen. Okay.
Yeah.
Because growing up for me without all this structure and stuff, ATP hydrolysis as an energy cofactor in making stuff happen was kind of magic. ATP is hydrolyzed and this happens. Okay. Inside this is actually mechanistic description of how that happens.
I didn't go into that.
So I have another impression that I'm on the technology that I want to check myself on, and that's the use of the cryo-EM. We've talked about this before. The solving molecular or atomic structures of proteins in the past involved crystallography, where you actually form a crystal, and that's a hit or miss process, and you get one structure, or maybe if you're lucky, you can engineer a couple of different structures, and it takes forever. This is cryo-EM, so you're looking at individual molecules that are just laid out on a grid, and you have it. My impression is that you have an assortment of different structures that represent different intermediates in this whole process that you can then, using computer technology, group the structures and say, oh, all these look the same, and all these look the same, and these look the same. So you have a series of structures that are all intermediates in this whole process, and then you can use, once again, computer assistance to put these together to in the appropriate sequence to show you what the pathway is. Is that, am I making sense?
Yeah,
Yeah,
that's right.
And not only is it that you have maybe different stages along the pathway, but how they land is random, and so that's part of the computational deconvolution of all the images. And, some of them are going to be useless because of which way they land. And so this whole concept of when you do this, you're getting an average, and that may have sometimes masked things like portal proteins and stuff like that.
Yeah, they use specific software to do that here, right? To look at the different populations and not have them all mushed together, and that way they can see the different states. Yeah. And they have names for them, which I don't remember.
Really remarkable. You can't make this stuff up.
But what's really remarkable, each human cell makes and consumes 10 to 100 million ATP molecules per second. The whole body, 10 to the 20th to 10 to the 21st molecules of ATP per second. I always am freaked out by the universe at the extent, but that's amazing. Per second, per cell. I can't even imagine how fast those motors are going, right?
Yeah.
Wow.
I just noticed something in one of these colored pictures that where they're showing their pentamer, and it has 5 different colors. There's 5 letters in Rep-40, so that's why they color Rep-40 with 5 different colors.
Yeah.
It's just really, really well done all the way around in the graphics for this paper.
Okay, now another really amazing paper. This one is in immunity. The title is Influenza, and I'm going to put virus in it. Influenza virus, they didn't, but the disease doesn't hijack anything. Influenza virus, it hijacks myeloid cells to inflict type 1 interferon-fueled damage in the heart. Oh my gosh, there are lots of authors here. I think there's just one first author, his first author is Jeffrey Downey, and then the contact is number 18. That is Philip Swirsky, and then we have correspondence also to Philip Swirsky. So this comes from Icahn School of Medicine at Mount Sinai, Mass General Hospital in Boston, Columbia University. I didn't know there was a Columbia Center for Genetic Errors of Immunity. Wow. Mount Sinai, Massachusetts General Hospital, Mount Sinai, and then Amsterdam University Medical Center. Oh my gosh, Kathy, you have a summary?
I do. And first off, I'll start with the fact that there's a commentary called a preview in the journal by Jana Cable and Jacob Yount. And I read it first as a preview, and I think that really helped me. And the title of that is Influenza Virus Hitches a Cellular Ride to the Heart. So influenza has been linked for decades to heart disease, and it's been detected in human, non-human primate, and mouse heart samples. And so there are a couple of big questions. How does it get to the heart? There's no multi-organ viremia or virus in the blood that would explain this, but somehow it gets to the heart. And once it's there, how does it cause heart damage? So they do experiments in this paper to address those questions. They first start off by further documenting the finding of virus in the heart. And they then get to some answers about how the virus gets there and how it causes the heart damage. And to make a long story short, a particular immune cell ferries the virus to the heart, where it then infects heart muscle cells that are called cardiomyocytes. Those heart muscle cells then get damaged by interferon-1 signaling that happens, and that negatively affects cardiac function. So they use special mouse approaches and models to show, for instance, that it's not just because the heart and lung are close together, and they use special mice to help identify the immune cell types. And this work is really highly significant. It suggests that the timing of therapy that you might give for cardiac complications from influenza, the timing might be important because the virus is cleared from the heart before there are any symptoms. And so just giving an antiviral might not work if the damage is due to this subsequent interferon response. And so they speculate on some ways that might happen. And overall, it may allow scientists to figure out a way to minimize heart damage from virus infection. This paper has a ton of data. There are 4 figures in the main paper, which you might think, oh, I can handle that. There are at least 25 panels per figure. And so that means there's over 100 panels of figures to digest from the main paper. And so also they're very small. So you pretty much have to look at it on a computer screen. And that doesn't include the supplemental figures, of which there are 6. And almost all of those also have about 25 parts to them. So about 250 figure panels to digest.
That's why there's so many authors.
Yes,
Right.
and I would recommend that anyone who is reading this paper have their browser window open to be looking up terms and sort of double checking on things, because there's a lot. This is a very dense paper in terms of the information that is in here and the things you sort of need to keep track of.
Yes.
But we're going to decondense it for you in a straightforward way, because that's what we do.
And I forgot to mention that they have a graphical abstract, but I much prefer the figure 1 of the preview by
I agree.
Cable and Yount. It's just bang on, simple, tells the story. So check that out.
I just tried to count them up. There's somewhere in the neighborhood of 50 authors.
Cool. Okay, so as Kathy said, there's lots of clinical data associating influenza in humans with heart problems, myocarditis, heart failure, and what they say, risk of myocardial infarction, risk of a heart attack. And that's because if you have serious influenza and you're hospitalized, you get better and they send you home. And then within the next couple of months, you have a heart attack. Same thing happens with COVID. I know this only because Daniel talks about it all the time. And Daniel says, it's the general nonspecific inflammations and the hypoxia that causes all that. You know, you have to breathe really heavily. You can't breathe. They just put stress on your heart. And they say that here. Maybe one of the mechanisms is nonspecific systemic inflammation that causes collateral damage. But they say it's not really well examined. So we're going to look at it here.
Right, because
And
they also say sort of that it could be the nonspecific systemic inflammation, or it could be a specific influenza impact on the heart. And so, is this just inflammatory disease and the heart seems to be susceptible, or is influenza doing something unique to the heart?
So they start by looking at autopsy records of people who died after laboratory-confirmed influenza virus infection, which they abbreviated as IV, unfortunately. I think IV is already taken. I think they should do IAV at least, which people use influenza A virus. Most individuals, no matter how old they are, have cardiovascular risk factors. They also got plasma from hospitalized influenza patients and looked for markers of cardiomyocyte damage. Troponin is a protein that you can monitor, which is a marker of damage, and also deterioration of cardiac function. And there's a peptide, N-terminal probrain natriuretic peptide is another one. They found that over 60% of the patients have signs of cardiac damage by monitoring these parameters here.
And starting to address that point that you were making about, is it just that general systemic inflammation, or is this unique? They do a control with patients who were hospitalized not with influenza infection, but with ulcerative colitis, which is also an inflammatory disease, and they don't see these types of increases. So that is sort of starting to tell us this might be something interesting and unique going on with influenza.
Right. So then they go to mice. They have a mouse model of IAV. Now they call IAV. And it's the H1N1 PR8 model. And they infect mice, and they have elevated troponin and NT-proBNP, those two proteins. They have cardiomegaly, and they have cardiac fibrosis. Then they measure the left ventricular ejection fraction,
In
the
a mouse.
LVEF, in a mouse. I don't know how they do that, but they measure it. So this is, how much percent of blood that's ejected from the left ventricle. And people, it's usually 50 or 70%. And below that shows you're having a heart problem. And so this declined in the mice too. Okay. The damage was dose dependent on the virus. It correlated with weight loss, which in mice, is a sign of influenza disease. But the mouse background didn't seem to matter. And they could see cardiomyocyte death, but nothing in other organs. This is funny. The colon got a little bit longer, shorter. Sorry, got a little bit shorter. Other comorbidities, aging, atherosclerosis, type 1 and type 2 MI, they worsened all of these parameters. And then, which I think is really important, vaccination protected the heart. Okay,
Yes.
folks?
What do you know?
protects
Vaccination.
the heart.
Really?
Another reason to get vaccinated.
Absolutely. Yes. The two parts of this paper that I was like most intrigued in wanting to go down a rabbit hole, and I have a lot of tabs of rabbit holes, are both supplemental figures, one of which is this one that I was so excited about, the impact of vaccination.
It's great. Okay, so then they say, okay, we see myocarditis, so let's take a look at inflammation. They see great inflammation in the lung, right? Monocytosis, neutrophilia, macrophage accumulation, but also bone marrow and blood. It still resolves 28 days post-infection. And the heart, a lot of myeloid cell infiltration. Macrophage numbers went up as well. And I don't know when they say this. It's just something that sticks in my head. All this happens after the most of the viral replication is over or starting to go down, right?
Yeah, they do mention that.
I think it's later. I just don't remember. They looked in other tissues for inflammation. They don't find any, really. It's just heart. And bone marrow, and blood, as you can see here. In the heart, they can see these infiltrating cells, the leukocytes, in focused aggregates randomly in all the areas of the heart they looked at, including the septum and the left ventricle. And then they said, we wondered whether, because the heart's next to the lungs, whether that had something to do with it. So they do parabiotic experiments where they sew two mice together, right, so that they share a circulatory system, and show that that's not the case. So they say this virus is somehow causing inflammation of the heart via the circulation.
Yeah. So basically, the idea with parabiosis is if something is transferred during parabiosis, that means that it had to sort of broadly circulate all the way around through circulation and can't just, traffic there other ways. I was thinking of very non-scientific ways to say that, but we'll just say traffic there other ways.
I would have liked to see some virus in the blood here, right? Because that's what they're saying is happening.
Not necessarily. They don't necessarily talk about whether there's free virus in the blood or whether there are infected cells moving through the blood. So there may be, so I think the idea is, there might be infected macrophages or some other infected cell moving through the blood. And it's not free virus.
Yeah, that would pick it up though, right? So I would still like to see that. And they can just do, they could do a PCR, they could do infectivity. Okay, so what's the cell type that's going on here? And here we go into lots of markers of different cell types. Hold on a sec. I lost my place here. Okay. So they do a number of approaches, which I don't know very well. So I'm just going to give you the summary of this. They're markers of. They want to know if the cells that are coming into the heart come from the bone marrow or if they're embryonically derived.
Yeah,
macrophages.
I'm going to phrase that a slightly different way.
Yes.
So I think of it as they're thinking about, are these macrophages that seem to be at increased numbers in the heart, are they macrophages that have gone through circulation, which we were sort of mentioning before, and then moved to the heart? Or is this a situation where macrophages that were already in the heart that are part of the heart tissue, so local tissue resident heart macrophages, are proliferating? So is it sort of trafficking in macrophages, or is it the macrophages that are already there proliferating?
Right. Got it. All right. So they use markers, cell surface markers, to look at that. And I'll tell you the conclusion is the macrophages that appear in the heart appear to come from the blood. Okay?
Yes.
Right.
But they also have weird markers.
Yeah. They have weird markers, and they don't fit into the normal places. But let me see if I can. So these, at 3 days post-infection, over 90%. So there's a marker of a certain kind of macrophage called LY6C, right? And they can have high or low levels on the cell surface. And the high levels are classical monocytes, is that right? And would those be the ones that are in the blood or in the the tissue?
So part of this is that they are using this label
Yes.
on the cells. So the cells have to go through a sort of set of developmental stages, and in order to make different types of macrophages. And it turns out that there are two different types of macrophages that come through slightly different developmental stages. There's sort of the more typical classical macrophages, as Vincent mentions, and sort of these atypical weird macrophages that are we think about less. And so they're using this fate mapping mouse where basically any cell that went through the normal pathway of macrophage differentiation, this is the normal pathway of bone marrow derived circulating macrophages, not the ones that lived in the heart.
Right.
They get turned a color, which I think is red, but I don't actually know. It's TDT plus here. In my head, I imagined it was red. So cells that have gone through that developmental pathway are red. So they see that in the blood at day 3, most of the cells are Ly6C high, but they also were red. They were also TDT plus. And so that means they went through the classical phase of development. So here, the classical part is coming less from the Ly6C and more from the fact that they turned on this marker and sort of became red, because at some point they had used whatever this other protein was during their developmental stage.
Right. So this is an atypical lineage, right? That's accumulating in the heart.
Yeah.
And they confirm it by a variety of markers and confocal microscopy.
Yeah, so the markers say it's not a cardiac macrophage. It's not a heart macrophage. It's one that infiltrated, but it's not one that went through the classical developmental pathway. It went through some other side developmental pathway because it didn't get this red thing turned on.
And they call them pro-DC3 DC3s. Okay?
Yes.
And the confocal shows these are interspersed with the cardiomyocytes in the infected heart, right? So they're right in there where the infection is going on. Okay. So let's talk about the virus now. So a lot of virus in the lungs, right? 10 to the 6 PFU per tissue. I don't know what per tissue means. It's usually
I didn't
weight.
know that. Confused.
Maybe there was a gram there. So clearly, they say the heart supports viral replication. They can see it in the aorta, the lung draining mediastinal lymph nodes, and the thymus, but no other tissues. At day 3, they can see this. And they also can look at transcripts of one of the viral mRNAs, NS1s. Only the lung, heart, aorta, and mediastinal lymph nodes became inflamed and supported viral replication. So they say maybe there's a link between the migration of these leukocytes and replication. Now, in the mouse, here's what I mentioned before. The titers drop after day 6 before any cardiac symptoms start. So the virus titers are starting to drop. In people, they do immunohistochemistry. They've got samples from people for viral protein, the nuclear protein. They can see it in the heart sections, so they're influenza positive cells in the heart sections in their people. So, in other words, virus is in the heart in mice and in humans. It looks like it's replicating there. So why is there so much virus in the heart, right? It's basically the lungs and the heart now that the virus is in. And they do, again, a parabiosis experiment showing that it's not just because the heart is next to the lung, and a lot of viruses is coming in there. So they say, well, maybe these leukocytes are bringing virus in. So they sort different populations by cell sorting from the blood and hearts of these mice. And the cells that have the pro-DCs and the CD64 macrophages in the heart contained viral NS1 protein. So they think that these LY6C high cells, which include the pro-DC3s, are accumulating in the heart. They differentiate into macrophages or DC3s, and that's where the virus is.
So it looks like those cells got infected in the lung and then somehow migrated to the heart.
So those cells, you said that they are not part of the typical evolution.
Development.
Development. Thank you. But that doesn't mean they're there normally, right?
Yeah, they're there normally. This is one of the other places where I ended up down the rabbit hole because I've always known that there's sort of certain types of heterogeneity between macrophages and dendritic cells, but I haven't always kept up with all of the different aspects of the literature. And so the classical is sort of the first pathway of development we described and the one that most people think of. and the one that we often sort of make assumptions is the one, but there are other pathways that we are determining that, macrophages and dendritic cells can come from multiple places.
Right. So these cells are a specialized cell
Yep.
that's there all the time.
Yeah.
They're picking up virus in the lung and somehow trafficking it to the heart.
Yep.
Right?
Exactly.
Okay.
So they also find viral nucleoprotein. They look for lymphocytes, leukocytes in different places. They find this nucleoprotein in these LY6 high cells. They find it in neutrophils and lymphocytes in the lung circulation. They also find this in the tissue parenchyma. In the blood, these nucleoprotein, influenza nucleoprotein positive cells are mostly CD319 positive. They also find NP positive LY6C high cells in the peripheral blood, but nowhere else. In the heart vasculature and the heart tissue, there are NP infected leukocytes, and in the myocardium, they're mostly CD319 positive pro-DC3s, this cell that they've been following. And they confirmed that by parabiosis as well. And they clear the lungs of infected mice. They can still see these cells in the respiratory tract and the parenchyma. And then the cleared hearts, they can see foci of infected cells, most of which express CD319. Okay, so looks like these cells are infected. These pro-DCs are infected. DC3s are infected. So they say, the chemokine CCL2 is important for mobilizing monocytes. So they said, is this important for recruiting these pro-DC3s to the heart? And they do a variety of correlative experiments, but one of them, I think, is really telling them. They looked at 19 different tissues, and the heart and the aorta are the most CCL2 rich at homeostasis, right? And then they looked to see if these pro-DC3 cells accumulate because of CCL2 and its receptor CCR2. They did adoptive transfer of the relevant cells. So they take bone marrow LY6C high cells, they purify those, they infect them in vitro, and they inject them into naive mice. They go to the heart, and they're mostly CD319 positive in the infected mice. If you transfer cells that lack CCR2, the receptor for CCL2, you don't have any of this cardiac phenotype. They also look at knockout mice or mice treated with an agonist, which inhibits CCL2 activity, fewer infected cells in the heart, lower cardiac viral loads, and low plasma troponin.
So just to sort of restate that, because I didn't get that through on the first read through, but the aorta and the heart are the most CCL2 rich at homeostasis. That just means at baseline.
Mm-hmm.
Right.
And then after infection, there's more transcripts and more protein abundance.
Right.
Yeah. So I would think about it that, the heart and the aorta are making a lot of this chemokine, CCL2. And so that's basically going to bring in any cell that happens to have the receptor. It's going to recruit any cell that happens to have the receptor. And when these pro-DC3s get infected, they seem to have this receptor. And so they end up going to the heart because that happens to be the place with the chemokine that matches the receptor they've just upregulated.
So,
and then if they use cells from mice that don't have the receptor, then that doesn't happen.
Yes, then they, as they say, accumulate elsewhere.
Okay.
So for myself and for the masses, I want to spend 30 seconds on chemokines. So these are proteins, right?
Mm-hmm. Yes.
That have the effect of attracting cells that have the appropriate receptor on them.
So
correct?
chemokine basically is chemotactic cytokine. So they're secreted proteins in the immune system that induce movement towards or away from whatever
Right.
stimulus.
And so you can think of it as if these are being made by certain cells in the heart, and they are extruded from those cells. The net result is a gradient of chemokine in the bloodstream or the tissues or whatever, and cells with the appropriate receptor can, in effect, smell that stuff and traffic towards the source of the smell.
Yes, there is a fantastic person online who makes immunology-related comics. And he has a comic that is related to this that I cannot, it's always in my head whenever I think about chemokine gradients. It sort of permanently shows up in my head, where he shows a cell making some chemokines and it says, "My chemokines bring all the boys to the yard." And so that's sort of the idea of what's going on here.
But we don't know necessarily why the heart is making this particular chemokine under these circumstances, but be that as it may, it has the effect of attracting these cells that just so happen to be infected with influenza.
Yeah.
Okay.
So also they look in the hospitalized patients, and there is a correlation between CCL2 in the blood and this NT-proBNP marker of heart damage in the hospitalized influenza patients, but not in the patients with, what was it?
Ulcerative colitis.
Ulcerative colitis. So again, this may be conserved in mice and in humans. So next they want to know what cell is infected. So they look and they find that, again, an NS1 protein of the virus is a marker for infection. They can find it in cardiomyocytes, but they don't find it in endothelial cells. They don't find it in fibroblasts. So only the cardiomyocytes seem to support viral replication, and they in fact die after replication.
I don't know why, but somehow that blows my mind. I don't think of cardiomyocytes as being a good home for something like flu, but I guess I'm just dead wrong. Okay.
A lot of ATP being made.
Yeah, and cardiomyocytes. Oh, good point.
It's true.
Cardiomyocytes. You kill them and they're gone. They don't necessarily regenerate, right?
I don't know. It's a good question. Are there stem cells that would regenerate?
I think that's right, but I'm not totally sure.
Well, Brianne's going to look it up.
Because my understanding is that when you have a myocardial infarction and you lose a lot of cardiomyocytes, they're replaced primarily with scar tissue.
Yes. Cardiomyocyte regeneration is very limited in adult mammals. Only about 1% can be regenerated, though it is higher in neonates and in other species.
So there are drugs developed by a number of companies that are trying to stimulate cardiomyocyte regeneration because it doesn't happen very effectively.
Right. So this is not something you want to happen because you're not going to heal from this very readily.
I think things should stay away from your heart. It's pretty important.
Yeah.
So far, I've learned from this paper, vaccination is good.
Yeah, exactly. That's right.
And I wanted to add the point that although they don't test it extensively, they do say that it can happen even if it's mismatched virus vaccine.
Yes. Yeah.
Yeah. That's pretty cool. All right. So then they would like to look at this infection. So they do RNA sequencing, single nucleus RNA sequencing, and they look at gene patterns that are being expressed. And what they find is a nearly ubiquitous increase of type 1 interferon. And then genes and processes involved in macrophage and monocyte correspond with antiviral and inflammatory responses. Okay, so interferon is a big one. So they do a whole bunch of analyses subsequently with markers and a program called CellChat. But basically, they think the viruses get transferred to the cardiomyocytes from these infected pro-DC cells, maybe by intercellular connections and apoptotic blebbing happening in the pro-DC3s, which can transfer viruses from one cell to another. So they then take cardiomyocytes from mice and they culture them, and they mix them with plasma from infected mice. They mix them with infected B cells, and they mix them with infected LY6C high cells. Okay, and that includes the pro-DC3s and other things. So the cultures with total infected LY6C high cells caused death of the cardiomyocytes, and they found infectivity in the function of PFU. And the ones they incubated with plasma from infected mice or infected B cells, nothing happened.
Yeah. So, I mean, that kind of implies there's no free virus, that the virus is traveling inside of the
for sure
319 positive cells.
Yeah. So it does imply, although I would have done a plaque assay on that plasma just to see, right? But so they say these CD319 positive pro-DCs, DC3s, transfer the virus to cardiomyocytes. It replicates, kills them. Boom. That's what's going on here. But that's not all. We want to know why they're dying, right? What's causing the damage? I mean, many papers would stop there because that's pretty important. So they said, well, the interferon goes up in our pathway analysis, so maybe this is killing the cardiomyocytes. So they take IFNAR, type 1 interferon receptor deficient mice, and when you infect them, the heart is protected. The viral loads actually don't go up. The heart's protected, reduced troponin, and better left ventricular blah, 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 LVEF after infection. Ejection function, I forgot that.
Ejection fraction.
Ejection fraction. But interestingly, if you treat wild-type mice, but not mice lacking the type 1 interferon receptor, with a double-stranded RNA, poly-IC, which is an agonist of TLR3, this accumulates in leukocytes in the heart. They release troponin, which shows you that just interferon is what is killing the cell. The virus is not doing anything on its own except triggering the production of interferon.
Yeah, and so I'm kind of like, how did this interferon get to the heart? And does any interferon lead to heart damage? And what's up with this? And so this is the other place, this like supplemental figure with this poly-IC leading to interferon damage in the cardiomyocytes that I am like, I want to know a million more things about this.
It's interesting because they treat patients with interferon, right? And so do they get heart damage? And they have actually used poly-IC in some trials to treat certain things. And does that have any heart problems?
And is this what's going on with those COVID patients that you mentioned that Daniel talks about? Is it not the general inflammation, but is it similar interferon?
It could be. It could be. It's very interesting. So the source of this interferon are these CD319 positive pro-DC3s. They're making a lot of the interferon. They tried to make mice lacking it and see if they could target the IFNAR1 on these leukocytes, but it didn't work. So then they tried silencing the type 1 interferon receptor on stromal cells, okay, and making chimeric animals. And when they do that, it didn't work either. But then finally they were able to target IFNAR on cardiomyocytes through a different approach. And then, so they're targeting the interferon receptor on cardiomyocytes now. And so forget about the producer cells. These mice do better with they have reduced troponin, better LVEF, and no off-target effects. So if you eliminate the receptor on cardiomyocytes, you protect the heart. That's the key to doing it. And so then they say, let's try a therapeutic approach. First, we're going to delete IFNAR on cardiomyocytes by CRISPR targeting, okay? And that works. And the second strategy is they express a dominant negative truncated IFNAR protein that sequesters interferon-1 and stops the signaling and ISG production. And this protects the heart also. So two different ways of targeting the IFNAR on these cardiomyocytes protects the mice. So that could be a therapeutic approach. Although I don't know when you would do it, right? If you have a hospitalized patients with influenza, you would do it on all of them.
There's
I don't know.
somewhere in here there is a correlation between cardiac complications from flu and pre-existing conditions. So I would say that you, if you were going to use this as a therapy, it would be targeted towards people who come in with pre-existing cardiomyopathies of one sort or another that are prone maybe to having a problem with an influenza infection.
So basically, the heart damage is because these circulating myeloid cells, they get infected. They go in to the heart. They're attracted there by CCL2. They transfer virus to the cardiomyocytes. It activates interferon, and that damages the cell. And you can interfere with that by targeting the interferon receptors on the cardiomyocytes. Amazing, really amazing, huge story, right?
Mm-hmm.
Now, the thing is that's really interesting is, so why is this happening?
Yeah.
Right?
Is there
So
some
they say
evolutionary drive to this, or is this just
so
happen?
they say by hijacking the specialized monocyte like pro-DC3, the virus preferentially accumulated in the heart, not necessarily because of intrinsic cardiotropism, but perhaps because the heart, for reasons we don't know, is a CCL2 superproducer. So it just happens to infect these cells that like CCL2.
Yeah. So
Wow.
what type of things do you actually want to traffic to the heart using CCL2? Sort of like, why did you evolve a lot of CCL2 there?
Yeah.
Also, why are cardiomyocytes, so susceptible to interferon mediated death? That also doesn't seem like a great thing.
Yeah.
Evolution wise for these nice precious cells.
They also say that you cannot use an antiviral because this happens when virus is gone pretty much, right? So vaccination works, or if that's not working, you can target the interferon receptor on cardiomyocytes.
So
Although I can imagine that's going to be kind of dangerous.
Yeah, that
right?
seems like there are a lot of other things I'd like to study before I did that.
yeah.
Kathy, you were going to say
Well, I was
something.
just thinking that, I mean, maybe an antiviral would be too late, but, who knows? Maybe it's a matter of amount. If you got an antiviral early, and so then there were fewer of those immune cells that get infected and go to the heart, then, maybe this wouldn't happen as much or to such a bad extent.
Yeah, they say here, as Rich said, the data suggests that the most severe manifestations of influenza occur in people with pre-existing cardiovascular disease. That said, the damage to the heart is not the result of infection. Even very high titers, viral titers, induce low and transient cardiac infection that peaks before symptoms occur. Rather, it's the festering interferon 1 response, not anti-interferon. There are a lot of people who have antibodies against interferon.
I know papers keep coming out about those individuals.
And I wonder
I'm always shocked
if they have less cardiac problems with influenza, right? I mean, their COVID is worse because the virus gets out of control. So maybe you never get to this. Maybe they die earlier. I don't know.
Yeah,
from
well,
virus.
I didn't actually really know that was a thing to have neutralizing anti-interferon antibodies until that group of COVID patients was reported. But I think I actually saw one earlier this week, and I have seen recently papers about pathology and patients having anti-interferon antibodies in a bunch of different situations. And so now there are a bunch of people who have been identified with those antibodies. You could really start to look at some of those things.
It's like 20% of people over 65 have antibodies to interferon. It's crazy.
Yeah.
Maybe there's something bad about interferon. Oh, it kills their heart cells. Maybe something's going on there. Wow, it's really interesting.
It's like the second time this week that I'm on a podcast with Vincent where I've thought about a scene in a movie where someone injects themselves with interferon and then has a heart attack.
Oh, yes, that was Dallas Buyers Club.
Dallas Buyers Club.
I didn't even remember that. It's interesting.
Matthew McConaughey, right?
Yes, yes.
But it should have been good for him.
It was not good for him.
Because according to this, it would.
Yeah, I think this is, among other things, a matter of dosage.
Yeah, I think that was
Interferon is potent stuff.
I think that was a too high dose all at once problem.
Yeah.
Wow. Anyway, I really love both of these papers. I think they're so cool.
Great stuff.
While we've been talking, I've been looking in more detail at these figures where instead of saying heart or lung, they have little colored images of heart and lung. And then as the paper goes on, I noticed, there's a brain and there's a kidney. And then there's somewhere it's just like different kinds of myeloid cells. And then I saw this icosahedral virus. And it's our friend AAV, which is how they use to make their tissue-specific knockouts using AAV.
Yep.
That's in figure
cool.
6N. And so some of the places I think they could have just used words, but
it's a massive paper and a massive work and really important.
Yes.
Really tells you why you get heart problems with flu. It's amazing.
Get vaccinated.
Yes.
Yeah.
Okay, let's do the couple of emails. There are only 3. We can get through these. Kathy, would you like to take the first one?
Sure. Sueran writes, Dear Vincent and Company, I was excited to hear that you combined 2 of my interests on the recent TWiV 1305, viruses and reptiles. I wanted to expand on a couple of thoughts voiced in the episode. Snake husbandry. It is not unusual for reptile enthusiasts to have a large collection of animals, and if they are private snake breeders, then this can easily extend into the tens or even hundreds of animals, often housed in tub and rack systems, especially when young. So viral transmission risk is amplified in such setups. Typical advice when bringing in a new reptile is to keep them in a separate quarantine room with dedicated equipment for anywhere from 6 to 12 months before introducing them into the main area housing the owner's other reptiles. However, if this virus can present asymptomatically for extended periods of time, then these quarantine periods will be less effective. Carpet pythons are a very popular pet here in Australia and overseas, both because they are fairly placid, don't get too large, usually under 4 meters, and have diverse natural patterning that has been exploited to produce a wide array of color variants. Australia banned the export of native reptiles in 1984, so assuming this virus is of Australian origin would suggest that the virus would have already had to have been transmitted to the wider world by 1984, or may have been carried into other countries by the illegal wildlife trade, which is unfortunately very active. A $600 lizard can, for example, sometimes fetch $50,000 overseas. Finally, the cheery virus name stems from the locale. The Sunshine Coast is a region north of Brisbane, and true to its name, has some lovely beaches. There is also a local university named after the region. Keep up the good fight, Sueran, who's a senior research fellow at the University of Queensland.
Sunshine virus. That's the name. I didn't know why it was called that. There you go. That's very cool. Thank you. Wow, $50,000 for a snake.
Yeah, really.
I wouldn't even pay $600 for a snake.
I would not either.
I don't think you should keep them. I think they should be living in the wild, but that's up to you. All right, Brianne, can you take the next one?
Sure. Molly writes, Hello, TWiV team. I've been listening to multiple MicrobeTV podcasts since mid-2018. As a bacteriologist, I was surprised but delighted to hear one of my papers introduced as the subject of TWiV 1295, published February 6, 2026, but I just listened to that episode this weekend. Thank you all for taking the time to read and discuss the paper. I'm writing to hopefully clarify a point and share some extra details that might interest you. First, you got the name right. We called it POKE because the model is that it is poking holes in the membrane. Excellent. I remember that, and I remember we were trying to decide if it was like how to pronounce it, and we liked POKE. Second, the group expressed some confusion about the idea of prophage competition, how POKE gives a competitive advantage to GIFC3, the prophage that encodes it, and how such a mechanism was selected. Without getting into the weeds too much, the idea is that even though both GIFC3 and BTP1 can induce expression of the GIFC3 lysis genes, only BTP1 activates POKE as a part of its lytic cycle. Therefore, GIFC3 can replicate unimpeded when it is induced, but BTP1 gets restricted. The key thing I think you are missing here is that both prophages need to be induced to replicate. They are not necessarily active simultaneously in the same cell. We can only see that the net effect at the population level is an advantage to GIFC3. The selective benefit of POKE is probably linked to phage defense rather than prophage competition because one of the prophages GIFC3 natively co-resides with activates POKE. The polylysogen of GIFC3 and BTP1 is a non-native combination of prophages, and the competitive advantage to GIFC3 is fairly small. The competition experiments here were mainly for proof of principle to show that POKE can affect prophage replication, even though it doesn't affect GIFC3. In contrast, in my first paper, I characterized an antiphage defense system and specifically defined it as a prophage competition element. This element, called remain, is encoded in one prophage and strongly blocks lysis by naturally co-residing prophages without any effect on the prophage that encodes it. It's called remain because prophages remain within the cell rather than being released by lysis. And she gives a link. Remain brings me to one small correction to the discussion. It was presented that the ATPase REM-S acts by depleting ATP. We actually do not know how REM-S blocks BTP1 or synergizes with POKE. Rather than just depleting ATP, ATPases are often activators of effector proteins and antiphage defense systems. For instance, REM-A of remain, the ATPase for which REM-S is named, activates a tRNase. Lastly, the group noted similarity between terminology we used and immunology. This language was intentional because this paper fits into the larger body of work that has identified many parallels between eukaryotic and prokaryotic immune systems. There are many phage defense proteins with strong structural similarities to eukaryotic immune components. The prokaryotic versions are believed to be ancestors to the eukaryotic versions in many cases. There is also functional similarity, which is the focus of this paper. As you mentioned, determining the quote-unquote right time to activate immune mechanisms is important because they are often toxic. Most characterized antiphage defense elements are activated by structural features of phages, i.e. capsid or tail proteins. These phage features are analogous to PAMPs in eukaryotic immunity. Fewer antiphage defense systems activated by the action of the pathogen have been characterized. This type of mechanism is analogous to effector-triggered immune responses that detect the effect of the pathogen on the host rather than a specific pathogen component. We use the term autoimmunity because prophages are phages and thus have a high potential to activate antiphage defense systems that they encode by either a structural feature or activity of replication. There are many different ways prophages avoid this scenario. Here are two great articles that discuss similarities between eukaryotic and prokaryotic immune systems. And she gives two links. Thanks again for discussing my paper, Best, Molly. And Molly is a postdoc at Princeton.
Nice.
Cool.
I love when authors pick up our discussions.
Yes, Molly, great paper. We're excited to talk about it.
I hope that Jolene sees this.
She will, but
does-
she'll be doing the timestamps.
Timestamps, yeah, hopefully. Rich, can you take that last one?
Sure. Simon writes, Dear TWiV, although I don't listen to all MicrobeTV episodes, we can quit reading now.
Yeah.
As much as I used to, I still listen as much as I can and still appreciate everything you all do. In TWiV 1305, there was some discussion as to how much lab leak conspiracy discussion is a thing these days. I'd like to suggest that it's still an important issue that absolutely needs to be tackled head on, and here is why. Anti-vax. A lot of anti-vax and big pharma conspiracy builds on the distrust of science and scientists. If you scratch those pushing and amplifying anti-vax, you will frequently find they also believe in lab leak. In fact, they must believe in lab leak, as their belief system is built on a need to fundamentally distrust scientists and their motives. Maybe someone should, I read this before. Here we go. Maybe someone should ask Alina Chan to review the paper you discussed. She often said that if the pandemic were ultimately proven to have a purely natural origin, she would feel very bad about the consequences of promoting the lab leak possibility. Keep up the great work. All the best. Simon. Well, somebody who is invested in lab leak is likely to twist whatever paper we might want to discuss to discredit it or whatever. So
this is
am
an
not
interesting
inclined.
idea that anti-vax and lab leak go hand in hand.
Yeah,
It's
makes perfect sense to me, yeah.
Mm-hmm.
But I don't know why they have to distrust scientists. It's only, I mean, it's mostly non-scientists that are portraying them as distrustful, right?
Right.
It's not right.
Sometimes I worry that it's just that like they're still sad about a grade they got in high school chemistry.
That's funny. That's really good. Well, this Alina Chan thing, so she knows that it will be nearly impossible to say, yeah, for sure we know it came from nature, right? But all the data we have says that, and there's no data that says it's a lab leak. But for her, it's not good enough. And that's a false bar to set up there.
Yeah, for some reason I was thinking about this, I think as I was studying for this TWiV, I was thinking that you won't find probably any scientists who will say under most circumstances that this or that is absolutely true.
Yeah.
And that's how you can tell it's a real scientist. If somebody says, this is absolutely the truth, you should be immediately suspicious. And when somebody says, well, could be this, could be that about some scientific thing, that should not be a cause for distrust. That should be a cause rather for trust, because this is somebody who's actually thinking about it and will accept that maybe it's not true, regardless of the mountain of data that says that it is true.
Yeah, I feel like when I talk about natural origin, I usually end up saying not, it's definitely natural origin, although it's kind of what I think. But I usually say the data that support natural origin are really good or are really strong.
Right.
Yeah, that's fine. But there are some people who say, I can't tell from the data where it came from, which is not right. I don't think that's fair, because there's a preponderance.
Yeah.
A huge preponderance. And no data that says that it came from a lab.
No, so that's not fair. Anyway, interesting idea. Okay, let's do some picks. Brianne, what do you have for us?
This time of year, I am spending lots of my free time thinking about March Madness and March Madness brackets. And there was an article in Scientific American earlier this week on the math of March Madness brackets, and mathematical ways that you can actually look at different brackets and actually use them to mathematically figure out teams that won, and all sorts of different math you can do looking at brackets, as well as the probabilities on perfect brackets with a perfect with random choice, or the probability of a random bracket getting a score of 0 points, and some mathematical modeling that people have done about this. So it says right now that for March Madness, the likelihood of getting a perfect bracket is between 1 in 8.9 quintillion and 1 in 9 quintillion.
Can't we just round it to 1 in 9 quintillion?
I think it is, but I also think that that's picking it randomly with no knowledge. There are some situations where you can have more confidence. I don't feel like all 9 quintillion possibilities are equally likely.
So we were having a family discussion the other day, inspired by March Madness, that my son-in-law brought up, and that's the Mexican food bracket.
Oh.
And you start out, who wins, tacos or enchiladas? Okay. And you go through this big thing. The overall winner was margaritas. Margaritas win every time.
Seems like a good bracket.
Yeah.
Kathy, what do you have for us?
Well, I picked two things. The first one, title of the whole thing is "Stuff Just Keeps Happening in Space," despite everything else that we do. Anyway, there's a SpaceX launch plume that has kind of a resemblance to a jellyfish, and it's really cool because of when it was launched and where the plume was relative to sunrise and so forth. And I just thought it was a really lovely apod, so I have the permanent link in for that. And then, of course, on Tuesday this week, there was a meteor that came in over Cleveland. Many people saw it and heard it. It was 2 meters in diameter, 6 metric tons, which is 6,000 kilograms or 6 megagrams. And it was just before 9 o'clock Tuesday morning. And people I know of in the Cleveland area heard it. There's a pressure wave equivalent to 250 tons of TNT. It traveled 34 miles above Northeast Ohio at 40,000 miles per hour. And so I included a link that has a map of where the meteorites were likely to fall, generated by NASA. It's a cleveland.com site. And so it also has a doorbell cam photo. Maybe, I don't know how many other photos there are of it, but there was that doorbell cam that happened to catch it. And the meteorites are probably pea-sized, but people are going to be out there looking for it. So something really exciting happened.
Oh, so nothing really big hit the Earth, huh?
It didn't hit the Earth. No, it burned up before that.
It burned up before it did?
But it's 2 meters in diameter and caused.
Can you imagine if that had hit
Yeah.
the Earth? Wow.
Yeah.
That'd be pretty bad.
Yeah.
Well, if they're a certain size, then they're not going to all burn up at some point, like the
Right.
one that killed all the dinosaurs, right? So,
Right.
wow. That's so cool that a doorbell camera picked that up.
Right.
Oh, my gosh. What randomness. That's so cool. Rich, what do you have for us?
I have something that has nothing to do with science whatsoever. But I was cramming for TWiV this morning, sitting in the living room with my wife, who reports to me things that I need to know out of the newspaper. And she picked this one out, which is just a nice, homey, feel-good story from the Washington Post there's this guy named Michael Coyne, who's 30 years old, who is autistic and has ADHD and is bipolar as well. He is supported by his mother, who started off raising him initially as a foster child and decided to adopt him. And when it came time for him to look for some sort of independent living, nobody would hire him. And so she created a business, a coffee shop, specifically for him that he could work in. And she and her family and other similarly disabled people run this coffee shop. This is just awesome. This story made me
That
cry.
already is great.
Yeah. And so the story is about one day somebody raided this guy's tip jar and took $20. And he noticed this. Good for him. And this really upset him that somebody, it's not so much the money, but you know how this is. You feel violated that somebody would rip you off. And this is in Warwick, Rhode Island. And word got out, including the mayor, and everybody chipped in and replenished his tip jar with $900.
Yeah.
So read it. It's a feel-good story.
Nice.
Good.
That takes care of the trash stories, right?
Yes.
Yeah.
All right. I'm trying to learn about space-time and curved gravity fields and stuff. I don't know why. It just is really fascinating. So
It's really interesting.
yeah, it's really interesting. And it's just, your world isn't what you think it is, you know? So I've been looking for videos. I've been reading Feynman's lectures in physics, which are really great. But I've been looking for videos too, and this one is pretty cool. This one is what causes gravitational time dilation? Why clocks run at different times, on the Earth's surface versus above. And, that's all because of Einstein's equation that we can actually correct the GPS satellites so that we don't get screwed up on the ground. Einstein's time dilation experiment. So this is an interesting, this is pretty technical. It goes into, so mostly you have to understand this stuff. You need to know math really well because there are a lot of formulas. And I look at these and I'm like, it doesn't mean anything to me. So they try and explain what this formula means, the time dilation formula. It's really long. And they try and explain the components, and I think it's really helpful. So it's by a channel called Dialect, which is all about different issues in science, I think. Clocks in a gravitational field tick more slowly, but why?
Cool.
That was Einstein. So the answers can only be extracted from within the abstract and insuperably mathematical depths of the infamous Einstein equation, but they have a simpler root. And it turns out that part of the Einstein equation has a few things from one of Newton's equation, which is really interesting. Newton was Einstein's hero, and so he was always trying to look at what he had done. We have a listener pick from Charles.
Wait, Vincent,
Yes.
before you move on,
Yes.
When you mentioned that and what you're trying to learn, part of the caption of the SpaceX jellyfish launch plume talks about the fact that it only looks like it's going down because the rocket follows the curvature of the Earth on its way to space. So that should resonate with you. I'm not
Yes.
sure it exactly resonated with me, but there you go. So.
Yes, it's following the curvature of the Earth not because Earth is pulling it. That's what Newton used to think.
Mm-hmm.
But the Earth is deforming the gravitation, the space-time around it, and therefore think of a trampoline and you put a big ball in the middle, which is Earth, and if you put a little ball around it, it's going to eventually circle around and go towards it. And that's kind of the deformation of space-time that's why it's curved. Yeah. I don't know it well enough yet.
That's about right.
Mhm.
as far as I understand it. I understand it like there, and then I still want to know why we live on a trampoline. And so
Well, the thing
that's
is-
about where I'm lost.
But Brianne, on the trampoline, if you put a little ball outside the Earth, it eventually spirals in and hits the Earth, which doesn't happen. So why does the moon stay at its distance? Is some other planet curving its thing? And I you know what I mean? I don't know why it's not falling in. Same thing with the sun. Sun has deformed the fabric of gravity throughout the universe, and so that's why the planets are all around it. But why do they stay in their orbits?
I think it's sort of like a conservation of momentum thing, but I'm not positive.
So the thing that Einstein really got was Newton said all the planets are held in place by the gravity of the sun, right? And he said if you took the sun away, they would immediately collapse. And Einstein said, no, there's nothing faster than the speed of light. So gravity can't be faster. It takes 8 minutes for light to get from the sun to Earth. He said, so it cannot be that the sun is pulling on the Earth. There's no magical pulling force. And that's why he said there's instead this fabric of space-time that's distorted by large objects. And it's so cool. It's almost like how many ATPs you make a second in a cell. It's that mind-blowing in my opinion. So biology can be just as mind-blowing as space.
10 to the 20th to 10 to the 21st ATPs per second per cell.
Yes,
That's
that's
what
what I
No,
wrote
per
down
body.
too.
10 to the 21st is the whole body. It's a 10 to 100 million per cell per second.
Okay.
Yeah.
Okay.
It's still a lot.
Got it.
I mean, I don't feel anything. I should be feeling a lot of energy, right?
You're warm.
Yeah, I am warm. We all are. Okay, we have a listener pick from Charles. Back to winter in Chapel Hill. Wet and 33, 1C, not my idea of good weather. Some of the recent digressions and picks reminded me of a couple of stories I read about 30 years ago. The link is to a video based on a short story by Terry Bisson. This is the version starring Tom Noonan and Ben Bailey. I hope you enjoy it. So this is a YouTube video called "They're Made Out of Meat," and I didn't watch it, but
I watched it.
What's
basically about a couple of aliens sitting in a diner, looking at all the humans around them and discussing about how amazed they are that all of this stuff they do comes just out of meat, that even their brains are made of meat and they can imagine stuff. And eventually
so the aliens are not made of meat?
Excuse me, I guess not. You never know. I mean, they present as humans, but I think that's just for the so that they can go incognito on Earth, and they decide that they've had enough of this. They can't handle it, and they say, "Well, what are we going to do with the humans that we brought up to our vessel?" And they say, "Well, we'll just do this or that to them, and they'll forget about it. Plus, nobody will believe them, and we'll send them back." It's very weird.
And that meat is making 10 to the 20th, 10 to the 21 ATPs per second.
It is pretty amazing.
That reminds me of To Serve Man. Do you remember that Twilight Zone episode?
Yeah.
You remember, Kathy, did you ever see the Twilight Zone? No?
No.
Twilight Zone was a favorite of mine, yeah.
1960s black and white Rod Serling kind of sci-fi, right?
Yep.
So this was about some aliens who come to Earth, and they go to the Congress and they say, "We have the plan to fix all your problems, shortage of food, energy, we're going to fix it all." And they have a book. And so everyone buys in, and they fix all the problems. The book is called
To Serve Man.
To Serve Man. And then they say, we're going to take a bunch of you now and show you our planet or something. And they take a bunch of humans, and they're basically going to eat them. So they just fatten them up to serve man.
Somebody figures out that the book To Serve Man is a cookbook.
It's a cookbook, yeah. So actually, Paul Offit did a Beyond the Noise last year, and he said RFK is, so the name of the alien was Kennebec, and he said RFK is Kennebec. He's coming to serve us all up to infectious diseases. All right, that's TWiV 1307. You can find the show notes at microbe.tv/TWiV. We'd love to hear from you. TWiV at microbe.tv. Comments, questions, picks. TWiV at microbe.tv. And we'd love your financial support as well. microbe.tv/contribute. Brian Barker is at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He is currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Kathy Spindler is professor emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun. These papers were just terrific. They were really fun to work on. I like
them. Yes. I have some more coming, too, that are equally good. Stay tuned. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.